Good morning. The topic for today is the regulation of gene expression. I'll be discussing this in two parts. The first part is mainly on the regulation of gene expression in prokaryotes. DNA, as you know, is the chemical vehicle of heredity. The functional units of DNA are called the genes. The genome represents the total genetic information in a cell. When you look at a prokaryote like E. coli, it contains 4,400 genes on a single chromosome. The genome of humans, they contain 23 pairs of diploid chromosomes, 6 billion pairs of DNA, and the genes are approximately 30,000 to 40,000 genes. Only a fraction of the genome is expressed in the case of eukaryotes. What is gene expression? Gene expression means it's a multi-step process that ultimately results in the production of a functional gene product. This could be either RNA or protein. The first step in gene expression is transcription, which is the primary site for regulation in both prokaryotes and eukaryotes. The ability to regulate this gene expression gives the cell control over the structure and function of the cell. It is the basis for cellular differentiation, morphogenesis, and the adaptability of any organism. Adaptability. What is adaptability? This is a process whereby organisms adapt to environmental changes by altering gene expression. This has been studied in detail in the prokaryotes, that is, in both bacteria and viruses, and involves a delicate balance of many processes. Now, when we look at the gene regulation, we find that there are 6 potential points at which the amount of protein can be regulated. The first site at which regulation could occur is the synthesis of the primary RNA transcript. So, the first site is at the level of transcription. The second site where regulation can occur is at the post-transcriptional processing of mRNA. Third site is at the site of mRNA degradation. Fourth is at the site of protein synthesis or translation. Fifth is any post-translation modification. And sixth is the site of protein degradation. The concentration of a protein is controlled by any or all of these points, and regulation at the level of transcription is the best documented one. Now, a specific protein may bind with certain regions of the DNA in the vicinity of the transcription site. This can have positive or negative effect on transcription. You call it a positive effect when expression of genetic information is quantitatively increased by a specific regulatory element, and this regulatory element is known as a positive regulator. You call it negative regulation when the expression is decreased by a specific regulatory element, which is called otherwise as a negative regulator. Now, originally the hypothesis was one gene transcribing or translating into one enzyme. This hypothesis is no longer valid, and it is replaced by one cistron, one subunit. What is a cistron? It is the genetic unit coding for the structure of the subunit of an enzyme or protein molecule. So this has been replaced by one cistron, which is expressing a subunit of an enzyme or protein molecule. Inducible genes. The response of the inducible gene increases in response to an inducer. What is repression? The gene products that decrease in concentration in response to a molecular signal is considered to be repressible. and the decrease in gene expression is called as repression. Housekeeping genes. Some gene products that are required all the time at constant levels. So a constant unregulated gene expression is called as constituted, and the genes are known as housekeeping genes or constituted genes. Regulatory sequences, they are sequences embedded in the non-coding region of genome. An interaction between these segments and the regulatory molecule, for example transcription factors, can induce or repress the transcriptional machinery. And this in turn will influence the kind and amount of products which are formed. These DNA sequences which flank a gene are called cis-acting, that is, they are found upstream to where transcription may occur, and they influence the gene expression on the same chromosome. They are known as trans-acting when the regulatory molecule transits through the cell from its site of synthesis to its DNA binding site. For example, protein transcription factor produced from a gene on chromosome 11 regulates a gene on chromosome 6. Well explained by this figure. When these regulatory elements are found close to the or flanking the site where gene expression occurs, then they are known as cis-acting. So you can see in the figure C, the purple one which is flanking the gene which is going to be expressed is upstream to that gene, and it's known as a cis-acting modifier. Whereas in the case of D, the figure D, you find that this modifier is synthesized at a separate chromosome. It transits through the cell, and its action is on a gene which is present on a different chromosome. Okay, so such a process is known as a trans-acting modifier. Okay, its synthesis is on a separate chromosome. It transits through the cell and exerts its effect on a different chromosome. Okay, so it is not flanking the gene as in the case of C, but in case of D, it is present on it is being synthesized on a different chromosome and is exerting its effect on allele 2, which is present on a different chromosome, and then it regulates the expression of that particular chromosome. Operon. The concept of operon was introduced by Jacob and Monod in 1961. An operon describes the complete regulatory unit of a set of clustered genes. An operon includes the adjacent structural genes that code for the related enzymes or associated proteins. So an operon consists of the structural genes that is coding for a particular set of enzymes or proteins. It includes the operator gene, which has overall control over the process of translation. It includes the regulator genes, which are regulatory proteins coded by them, and it includes the promoter site, which is present between the regulator gene and the operator gene. So you're well aware of the lac operon. That is very important in the case of prokaryotic regulation. It contains the genes for the use of lactose as an energy source, and it is an inducible catabolic operon of E. coli. The structural genes for the lac operon are the Z, Y, and A gene, which codes respectively for beta-galactosidase, which hydrolyzes lactose to glucose and galactose, permease, which transports lactose into the cell, and thiogalactoside transacetylase, which acetylates lactose. The structural genes Z, Y, and A transcribes a single large mRNA, which is known as a polycistronic mRNA, with 3 independent translation units to form 3 distinct enzymes. The genes are coordinately controlled, and it's like a switch. It can be turned on and off as a unit. So if it is switched on, all the 3 structural genes Z, Y and A will express its proteins. If it is switched off, then all 3 enzymes are not being produced. So this is the structure of the lac operon. You can see the structural genes Z, Y and A, which is transcribing and translating into the enzymes beta-galactosidase, beta-galactoside permease and the beta-galactoside transacetylase. We have the operator or O gene located upstream to the Z gene. You have the regulator or I gene located again further upstream to the O gene. It transcribes the mRNA for repressor protein. And we have the promoter site just adjacent to the O gene, where the enzyme RNA polymerase would bind to transcribe the Z, Y and the A genes. The regulator I gene is a constitutive gene. It is independent of inducer and is expressed at a constant rate and leads to the synthesis of a tetrameric repressor protein. The repressor protein binds to the operator gene, and this prevents the binding of RNA polymerase to the promoter site, preventing transcription of Z, Y and A genes. The repression of lac operon in E. coli occurs in the absence of lactose. So this figure gives you very elegantly, you can understand what happens in the case of the lac operon. I told you the regulatory gene is a constitutive gene. It is continuously producing the mRNA which will then transcribe and translate to the repressor protein, the formation of the repressor protein. This active repressor will then bind to the operator site and this turns off the operon. And this occurs when the lactose is absent. When the repressor protein is bound to the operator site, then the RNA polymerase cannot attach to the promoter site. And the synthesis of the 3 enzymes which will be transcribed or translated by the Z, Y and A gene will not occur. What happens when lactose is added? So lactose can be considered as a gratuitous inducer. This results in de-repression of the lac operon. So it becomes an inducer molecule. It reacts with the repressor which is attached to the O locus or in the cytoplasm. It brings about inactivation by changing the conformation. DNA-dependent RNA polymerase can now attach to the P site and permeate to the operator site, which is now free, and transcription of the structural genes would then occur. So again, this figure gives you very elegantly, it describes what happens when the lac operon is de-repressed. So in the first case, we find that I told you that the repressor molecule is being continuously produced and will bind to the operator site and prevent transcription when no lactose is present. So without, the first case is the case when there is no inducer. In the second case, what happens? This is in the presence of an inducer like lactose. What happens? The inducer will now bind to the repressor protein. It changes its conformation and it is therefore unable to bind to the operator site. So now RNA polymerase can now bind and bring about the transcription and translation of the Z, Y and A gene. So what is a gratuitous inducer? They are structural analogues of lactose. For example, isopropyl thiogalactoside, IPTG, which can induce the lac operon. Unlike lactose, it is not a substrate for hydrolysis by beta-galactosidase. So this de-repression of the lac operon can either be brought about by an inducer like lactose or by a gratuitous inducer, which is a structural analog of lactose, like isopropyl thiogalactoside, or which is also known as allolactose. It can act as an inducer. It can bind to the repressor protein. It can change the conformation of the repressor protein, converting it to an inactive repressor. So the operator site is free. The RNA polymerase can now bind and transcribe and translate the three genes, the Z gene, the Y gene, and the A gene, giving rise to the three enzymes beta-galactosidase, permease, and trans-acetylase. Now there is also another process of regulation which is brought about by the catabolite gene activator protein, or the CAP protein. This CAP protein is a dimer. It acts as a positive regulator of many catabolic operons like the LAC operon. The attachment of the RNA polymerase to the promoter site requires the CAP bound to cyclic AMP. Now, when there is a decrease in glucose levels, it activates the enzyme adenyl cyclase. This adenyl cyclase catalyzes the synthesis of cyclic AMP. The cyclic AMP binds to CAP protein to form a CAP cyclic AMP complex. This complex will now bind to the promoter site and initiate transcription in the absence of repressors. Now, this figure gives you very elegantly the role of the CAP protein. Okay, when the glucose levels are low, then what happens? Cyclic AMP is formed, and this cyclic AMP will now bind to the inactive CAP protein, converting it to the active CAP protein, which then binds to the promoter site. Okay, so in this situation, that is in situation A, lactose is present, glucose is scarce, cyclic AMP levels is high, so there is abundant LAC mRNA synthesis. Okay. The repressor is in the inactive state, inactive LAC repressor. The RNA polymerase can bind, it can transcribe the translate and form the three enzymes. Okay, that is beta-galactosidase, permease, and trans-acetylase. Situation B, what happens? Lactose is present, but glucose is also present. So when glucose is present, cyclic AMP levels are low. So there is no complex between cyclic AMP and CAP protein. So the CAP protein remains in an inactive state. Okay, there is little LAC mRNA being synthesized. Okay, and the RNA polymerase cannot bind, and so the three enzymes will not be produced. This is because glucose is also present along with lactose. Since glucose is present, the nutrient is available to E. coli, and so it does not need to utilize lactose, and therefore the enzymes for breakdown of lactose is not being synthesized. Okay. So when only glucose is available, the LAC operon is repressed, and repression is mediated by binding of repressor to the operator site. The operator site has got this typical motif of helix turn helix, and it blocks transcription of the three enzymes which are necessary for the breakdown of lactose. Okay. Now, when only lactose is available, the nutrient for glucose, which is necessary for the proliferation of E. coli, is not there. So it has to make use of lactose, since only lactose is available. So for that, the LAC operon will get induced. A small amount of the lactose is converted to its isomer allolactose, and then it will act as an inducer. It binds to the repressor protein, changing its conformation. In this repressor protein will no longer bind to the operator site. as the glucose levels are very low or absent, cyclic AMP is produced, which binds to the cap protein, forming the cyclic AMP cap complex. This is activated. It binds to the cap site and the transcription is initiated. Now, when both glucose and lactose are available, the transcription of the lac operon is negligible, even if lactose is present at high concentration, since the organism can utilize glucose as its source of fuel or energy or nutrient. The adenyl cyclase is inhibited in the presence of glucose, and this is called as catabolite repression. No cyclic AMP cap complex is formed. The cap site remains empty. The RNA polymerase is unable to initiate transcription. So this is the regulation of the lac operon has been can be elegantly shown in these different conditions. So in the first case, that is, there is excess lactose, glucose is absent. So the natural nutrient glucose is absent, it has to make do with lactose, and lactose is present in excess. It will stimulate the lac operon to make the three enzymes, beta galactosidase, permease and transacetylase. Now in the second condition, we have excess glucose and excess lactose. So since glucose, its natural nutrient, is present in excess, it does not need the lac operon to operate, so the operon is turned off and the cap protein is also not getting bound because cyclic AMP is not formed in the presence of glucose. In the second condition, the third condition, you have excess glucose and absent lactose. So in this condition, since the natural nutrient is present in excess, the repressor protein, lac repressor protein, gets bound, the operon is turned off because the lac repressor is bound, and the cap protein is also doesn't get bound. In the fourth condition, there is absence of glucose and absence of lactose. Since both the nutrients are not available, then you have, even if cap protein binds, the operon will be turned off and the lac repressor will also get bound, the operon will get turned off. Now in the last condition, that is, there is absence of glucose and excess of lactose. Now the natural nutrient glucose is not available. The only available source of nutrition is lactose for the E. coli. So it has to make do with the lactose. For that, the operon is turned on. You find that the cap protein is bound. The repressor protein does not get bound. So the RNA polymerase will get bound to the site and the RNA will get transcribed. So these are the different conditions in which the lac operon gets turned on or it gets turned off, depending on the availability of whether it is glucose or lactose. Now transcriptional regulation is primary. That means regulation at the level of transcription is the primary site of regulation. However, regulation can occur also at the level of ribosomal RNA and protein synthesis. And this also plays an important role in the case of microorganisms, which tries to adapt to environmental changes. Now, there's a terminology known as stringent response. Now, E. coli has several operons synthesizing ribosomal RNA needed for the assembly of the ribosome. Each is regulated in response to changes in environmental condition, and regulation due to amino acid starvation is called as the stringent response. Now the operons for ribosomal proteins, that is the R proteins, can be inhibited by excess of their own proteins. Each operon has one R protein which functions in the repression of translation of the polycistronic mRNA from that operon. Okay. So in addition to the regulation at the level of transcription, you also see how the ribosomal proteins also play a role in affecting the translation of the mRNA and the formation of the proteins. Thank you.